All right, please welcome the William Monroe High School, Greene County Arabidopsis group.
Hello, everyone. I'm Jared. This is Becca, Danny, Joseph, and Lauren. Arabidopsis thaliana is often used as a model organism in biology and is widely studied by scientists to provide general information about plant genetics and their defenses against pathogens. Genes are also easily manipulated within Arabidopsis plants, making them spectacular research plants. In the case of this study, the APT2 gene is being removed from these plants. As of now, little is known about this gene and how environmental factors will affect the plant with this gene absent. What scientists do know is that it is responsible for amino acid transport throughout the clone. The HPA pathogen. So the removal of this would decrease the effect of the pathogen. The class tested the gene and copper sulfate in the soil, reporting data regarding leaf color, rosette diameter, and number of seed pods. Scientists worldwide have done countless experiments on the effects of copper sulfate in plants, but little is known about how it affects the growth development of Arabidopsis plants. Copper sulfate is known to stimulate the growth of plants, create a nitrogen deficiency, and reduce mold growth. The first hypothesis regarding the APT2 gene was the following. If the APT2 gene is removed from the mutant plants, then the wild type control will have greener leaves than the mutant control. The wild type control will have a larger rosette diameter than the mutant control. The mutant control will have a larger bolt height than the wild type control. The wild type control will have fewer trichomes than the mutant control. The wild type control will have fewer seed pods than the mutant control. The wild type control will have a longer reproductive cycle than the mutant control. And the wild type control will have fewer flowers than the mutant control.
The second hypothesis related to what we think would happen when you added copper sulfate to the plants, and it is as follows. If copper sulfate is added to the Arabidopsis plants, then the wild-type control plants will show signs of being healthier than the wild-type experimental. Such signs may include greener leaves, taller bolt height, greater rosette diameter, more seed production, more trichomes, more flowers, and we expected the wild-type control to have a longer reproductive cycle than the wild-type experimental.
The next hypothesis refers to the outcome of the plants without the gene that will be given copper sulfate. The hypothesis states, if copper sulfate is added to the mutant form of Arabidopsis plants without the AAP2 gene, then the mutant experimental group's leaf color will be more yellow. It will have a smaller rosette diameter. Also, the mutant experimental group will produce a shorter bolt height than the wild-type control group. Regarding the trichomes and seeds, the mutant experimental group will not exhibit trichomes and will produce fewer seeds than that of their counterpart, the wild-type control group, which will exhibit trichomes. Next, the mutant experimental group will not have a reproductive cycle, whereas the wild-type control group will. And finally, only the wild-type control group will present flowers and survive the 8-week-long life cycle. Sprouts appear on day 4, and rosette diameter was first measured on day 11. As the experiment progressed, there began to be a measurable difference between the wild-type and mutant rosette diameters. Within those groups, there grew to be more difference between the control and experimental rosette diameter. This can be seen on day 33 between the wild-type control and wild-type experimental rosette diameters. As you can see, there is much variance within our class of the rosette diameters. This variance is represented by error bars. Error bars are used to show the variance of data and to indicate the error or uncertainty of a reported measure. On average, the wild-type plants had larger rosette diameters than the mutant plants because of the removal of the AAP2 gene. The removal of this gene caused decreased efficiency in transporting amino acids and nutrients between the leaves and the phloem. This decreased efficiency caused negative health effects, such as deficient rosette diameter and the distinct presence of anthocyanin in the leaves and stem. The experimental plants, when compared to their control counterparts, had smaller rosette diameters. All of the plants had a similar life cycle length, so the discrepancy was likely due to the copper sulfate. The experimental plants showed yellow leaves, which may have been due to decreased photolysis, or not enough light. However, the yellow leaves may also have been due to the sulfur in copper sulfate, which is known to cause nitrogen deficiency. Nitrogen deficiency can cause yellow leaves and stunted growth, both which were observed in experimental plants.
Once a plant bolted, its rosette diameter was no longer recorded. This was done because when a plant bolts, it indicates that it is no longer investing the majority of its energy in widening rosette diameter. The mutant control plants bolted first, and the mutant experimental plants followed only a few days after. The wild-type plants bolted around 14 days later, and for the rest of the experiment had shorter bolt times. The accelerated life cycle in the mutant plants is an indicator of high stress. These high stress levels, like the reduced rosette diameter, are likely due to the removal of the ACT2 gene. The experimental plants had shorter bolt times than their control counterparts, even though they bolted no more than a week later. This was likely due to the copper sulfate. However, the copper sulfate did have a stronger effect on the mutant experimental than the wild-type experimental. Mutant controls had significantly taller bolt times than the mutant control had a significantly taller bolt times than the experimental. Up until day 51, the one-group measuring error caused a drop in the class average. While the wild-type control did have a higher bolt type than the wild-type experimental, it was by a much smaller margin than the two mutant plants. The stunted bolt height in the mutant experimental was thought to be a combination of the removal of the ACT2 gene and the addition of copper sulfate to the soil. However, on the graph to your right, the data was localized and the variable of time removed. After observing this data, it became apparent that the ACT2 gene did not affect the rate of bolt growth or bolt height, but only when the plant bolted. This indicates that the amino acids transported by the protein the ACT2 gene codes for do not build a protein that affects structure, but rather reproductive cycle length. In future research with protein, the amino acids transported should be identified to better understand and test the effects of the ACT2 gene on the Arabidopsis plant.
Mutant plants had produced significantly more seed pods than the wild-type plants because the mutant plants had higher stress levels caused by the removal of the ACT2 gene. Instead of focusing on its individual health, the mutant plant decided to focus on reproducing and passing all of its genes on to its offspring. For the mutant control, they had produced notably more seed pods than the mutant experimental. The wild-type control had produced more seed pods than the wild-type experimental up until the last 3 days, which is when experimental error caused more data to be entered into the wild-type experimental group. Overall, it seemed as though the experimental groups were attempting to produce as many seeds as the mutant groups, given that their life cycle lengths are similar. However, due to nutrient deficiency, they were unable to. Now, when you take away when the seed pods first appeared, and just focus on the amount of seed pods produced, it is similar to before. The mutant plants produce the greater amounts of seed pods, not because they are healthier, but because they are under stress.
Several changes in our experimental design could have produced different results. The plants received light through a suspended time life, which, given that the plants are near windows and the sun is inconsistent, is not a very accurate constant. If the plants had been planted inside of a light box where lighting can be completely controlled, the possibility of photolysis affecting the leaf color would have been eliminated. The fertilizer used to prepare the soil had equal concentrations of nitrogen, phosphorus, and sulfur. In the same experiment, if a fertilizer with more nitrogen and sulfur had been used, it is likely that the differences between the experimental and control plants would have been non-existent. Finally, the Arabidopsis plants were grown in a relatively unconstant environment. If they had been planted in a controlled environment, such as a greenhouse, the results produced would have been more accurate. Nonetheless, the results were very defined. The removal of the ACT2 gene is unhealthy for the Arabidopsis plants, and the addition of copper sulfate to the soil causes a nutrient deficiency that is also unhealthy for Arabidopsis plants.
To conclude, although there were certain parts of each hypothesis that were supported qualitatively and quantitatively, some elements were not supported, therefore all three hypotheses were rejected. The copper sulfate was harmful to the health of the plants, and removing the gene also stressed out the plants. Human error could have certainly caused an impact in the experiment by creating errors in the data. For example, one plot of wild-type control plants were unintentionally aerated. This resulted in the death of a few sprouts within that plot. Also, there were two occurrences of the experimental variable copper sulfate being added to the control plants. There was no noticeable change from this error. As a class, there may have been a slight inconsistency in data collection, specifically in the counting of seed pods. This may have led to an incorrect number of seed pods on the grass. The conditions that the plants were placed in may not have been ideal for their health. The blinds did not completely block the sun, so some of the plants may have been impacted by photolysis. This complicated the interpretation of the discoloration of the leaves. More specifically, it made it hard to determine which variable was impacting the color the most. Additionally, the windows did not always remain closed, so some insects may have gotten in and impacted the Arabidopsis plants at any certain point in their life cycle. Although both of the experimental groups responded somewhat similarly to the copper sulfate treatment, they definitely showed decreased signs of health and success overall, so therefore the copper sulfate treatment would not be wise to use on a larger scale. Also, we noticed a lot of decreased health in the APP2 mutants, the ones that didn't have the gene. So we don't think that it would be wise for a farmer to remove this gene in an effort to combat HPA. Finally, we would like to thank Dr. McDowell, his lab, and Virginia Tech for providing us with this astounding opportunity. Also, we would like to thank Ms. Dexter's amazing leadership along with the Blue Ridge Virtual Governor School. Thank you.
Dr. McDowell, or Chief?
You did a great job overall. What I really liked was how you reflected critically after the experiment was done about what you could improve. This is something that my group does all the time. You know, we're constantly thinking about, okay, are we doing these experiments the right way? How could we make them better? So that, kudos for that. Also like how you took a critical look at the way you were analyzing the data from the bolt height and seed plot and identified timing as the really key difference. That's a good insight. I do have a question for you. Your treatment contains two compounds with potential biological activity, the copper and the sulfate. Do you have any thoughts on which one was causing the effects that you saw between the control and experimental APP2 mutant?
Based on our research, we found that it was most likely that sulfur and copper sulfate was causing differences because it can cause nitrogen deficiency, that, and those were the main symptoms like that we saw as differences.
That's a great answer. I think we can test that critically because other groups they have sulfur as their treatment as well, but with different cations. So we can look for similarities there. Well done.
All right, who's got the microphone? Who's got the clicker? Our next presenters are Gooseland Block 1. Please give them a warm welcome.
Hi, I'm Taylor Teague. This is Alex, Delon, and Riley. So last time we were gathered here, our class decided that we were going to test the effects of adding nitrogen to the soil of the Arabidopsis thaliana plant. Our hypothesis was this: if we added 5 times the amount of nitrogen that is normally found in plant fertilizer to our plants, then in the case of the wild type plant, it would aid the plant and help it flourish and grow. But in the case of the mutant plant, because the aptitude gene affected the phloem, the nitrogen would not travel to the plant as efficiently, and it would not aid the plant as much. We measured the plants by using a metric ruler, and we measured the width of the rosette and the height of the bulb.
So, starting off this project and research, we had the help of one of our chemistry teachers at Gibson High School, Casey Rogers. She came in to help us and aid us make the 2-mole solution of KNO3. By doing this, we took 202.22 grams of KNO3, potassium nitrate, therefore adding it to 1,000 milliliters or a liter of water, mixing it around, creating a clear solution.
The control group was watered with distilled water every Tuesday and Friday. It is watered with distilled water to keep out any extra nutrients from getting inside the plant. The control group had 2 forms of the APT2 gene, which was in the wild type and the mutant plant. In the wild type, the APT2 gene had no modifications to it, and in the mutant plant, it was cut off. Our class observed that the control group lived the normal life cycle of the Arabidopsis thaliana, in which it grew, became stressed, and shortly after died.
So, as you see the graphs here today, this line graph shows the average rosette width of the plants. And so our experimental group was treated with 1.5 milliliters of the solution every Friday for about 5 weeks. And this solution was added via pipette. And after that, it was then treated with 1,300 milliliters of water in distilled water. So by this graph, as you can see, the experimental group grew a rosette width and the control group. However, the mutants actually wild type better the wild type. However, the wild type didn't pass it. But as you can see, the data does stop for the experimental group. Therefore, because the plant was starting to die quickly. Now, this graph shows the bulb height of all these plants. And if you look, you can see that the control groups were the only ones who really produced substantial data to the bulb height. And this control group, therefore, with the mutant in the red, you can see that it grew substantially greater than the wild type. And if you look at the experimental group, the mutant did the same thing as it did for the wild type. It grew greater in its height, but they still died at an early rate that we have found some data to support. And here you get the solution. We calculate the sleek is otherwise known as seed pods. And you only see about 0.5 sleeks on the experimental mutant because, as I said, these plants are dying quicker than expected. And however, the wild type did not grow in sleeks at all. It died quicker than the experimental plants in that, the mutant plants in that experimental group. But as you can see, the control mutant grew a greater number of sleeks and seed pods than the control wild type.
There are 2 sources of error in our experiment, which were the sleek number and the humidity levels. The sleek number was a source of error because 2 groups in our class did not record their sleek numbers, which slightly messed up our class data. The humidity level was an error because at Goochland High School, the air conditioning broke for about a week, which increased the humidity levels inside the classroom. We don't know if this had an effect on the plant or not because we were unable to control it. So in this picture right here, you see the wild type control plant. Now this plant right here was a great source of odd data for us. In this picture right here, which was taken on October 20th, the plant looks very stressed, has little bulbs, and the soil is very dry. Now this picture right here was taken a week and a half later on October 27th. You can see there is a drastic change in the plants. The picture right here is flourishing now, has flowers, bulbing, and the soil is very moist. We're unsure of why this is the case because we did water these plants twice a week for over the course of 4 weeks with only distilled water.
So one other source of air that possibly could have affected the data of these plants. In the beginning of this project, we actually tried tilting the plants to use less water throughout, so we can conserve all the distilled water, so we can have enough throughout the entire research period. And therefore, we overall concluded that it did not have an effect on the plant's growth.
At the beginning of the experiment, we thought that the potassium and the potassium nitrate would not have an effect on our plants. As we look back at our experiment, we now think the potassium may have had an effect on our plants. In solids, there's a sodium-potassium pump. We think the potassium may have clogged inside of the pump, depriving the plant of other nutrients it needed to survive. If you look at the picture right here, it's the sodium-potassium pump. The potassium went through the pump and into the top of the plant, where it let the plant flourish during the first treatment. As treatments continued, the potassium began to build up right here, thus clogging the sodium-potassium pump, and no other nutrients that the plant needed to survive were able to get to the plant.
So here, this is a picture of one of the experimental plants. As you can see, it does not look very good. Due to the fluorosis, you can see the yellowing and the browning of the plant, and you can also see a little bit of algae starting to form underneath the plant. But however, you can see, as we pictured in the rosette graph, the experimental group grew greater rosette and leaf structures. And this is partially due because of the excess nitrogen. It's been proven that excess nitrogen actually benefits the foliage growth of the plant, and therefore producing greater rosette and leaf structures. However, this increases the surface area, the color of the plant, and therefore it increases the photosynthesizing capabilities of this plant. And if you can photosynthesize more, it takes more energy from the plant to do that. Therefore, the plant takes all that energy from its other resources and reverts it into photosynthesizing and maintaining this foliage growth. Therefore, the plant forgets about having to go through the reproductive cycle and create siliques and mold.
As we all know, nitrogen is one of the essential elements for life here on Earth. It's used in the compound of chlorophyll, which then aids photosynthesis. In photosynthesis plant, energy from the sun is in the leaves, then transferred into food. All of the research here today presented will help Virginia Tech aid and refine the distribution of food all across the world. Who here likes apple pie? Or maybe Italian dinner or some hamburgers? We all like food, which is awesome. But unfortunately, some people can't say that. In the years of 2014 through 2016, it was found that 795 million people were malnourished. That's 1 in 9 people. Every year, it is projected that 900 million people die from hunger or hunger-related diseases. And the fact that touched me the most and reassured myself that I'm fortunate is that every 10 seconds, a child dies from malnourishment. These two men, Dominic Barrett and Steve Miles, are local Goochlanders from our home county. They created an organization called Shalom Farms in Goochland with all of the rural resources to help provide healthy and fresh foods for low-income areas of our home capital of Richmond. These two men founded their company in 2008, and in 2011, with the help of all of their volunteer farmers, created 35,000 pounds of food to distribute all across Richmond. These two men and many other groups and many other farmers across our counties can use our research found to help aid and diminish malnourishment across the country. To take it on a national standard, in 2012, all of the agriculture taxes were collected and were added up to be $390 billion. Agriculture is a big part of our economy. and out of that, $220 billion were for crops alone. It was not expected for our experimental group to shrivel up and die like they did. It is expected for the experimental group to live longer and flourish. Our hypothesis was somewhat correct as we look back at our experiment. It was somewhat correct because after the first treatment, our experimental group did live longer than the control and did flourish. But as time went on, this is how our hypothesis is incorrect. The plants began to shrivel up and die.
So, to relate this to all the topics and overall farming throughout the entire world, as you know, recently pesticides and herbicides have been shown risky factors to the plants and also risk to human health. And this means they've been banned in lots of regions across the world, so farmers have to find alternatives to treat their plants. And we know that herbicides help plants grow better, greater crop yield, and help fight off diseases. So this is why we chose the topic of trying to add nitrogen, because nitrogen is a key component of metabolism and photosynthesis in a plant. So if we increase that, we were hoping to find that this would create a greater crop yield and naturally help the plant fight off HPA, therefore helping it fight off natural diseases naturally through its own metabolic process and immune system.
As we conclude, we wanted to thank Dr. McDowell for answering our many, many emails that we have sent him, and for taking time out of his day to FaceTime us to make sure that we had everything to the standard that tech has. Thank you.
Any questions?
So those two pictures you showed were the same. That was two different groups, or that was the same groups?
Yes, actually, that was my group's plants right there, and we just came back one day and they were just like resurrected, and we had no idea what happened because we only watered them with distilled water. And
Yeah,
our
ours
plants
was
were struggled. Yeah, this was 1 week, and then 1 week later, we called them Lazarus plants. We called them Lazarus plants because they died and then came back to life.
Let's see the light. That was another really good job. One thing that I really appreciated was how you made the link back to agriculture. It's, you know, that's what gets guys like me out of bed in the morning, is, you know, the hope of eventually doing something that has at least a small impact on our food supply. So I appreciate that connection. Like you guys, I was really surprised at how quickly the experimental plants keeled over. Did you get, and you know, I know that you had a really short time frame between when they were looking great and when they were dead, but did you get any inkling of whether the mutant plants were either more or less susceptible to that stress than were the wild type?
Like, mutant plant?
Yeah.
Okay. So as I was showing you the graph, you can see that the mutant plant actually live longer and survive longer than the wild type. And
Right.
this is the possibility that since the excess nitrogen and the active gene in the mutant is cut off, the excess nitrogen possibly didn't have the time to travel all the way through the plants and it spread all throughout the plant. Therefore, it having less nitrogen, therefore the plant can survive longer and not have to process all the nitrogen all the time.
Yep, that's a really good explanation. I'll buy that. Now, okay, one last question. Did the time at which the death occurred, did that coincide with the air conditioner breakdown that you guys mentioned?
No, I think they started to wilt and everything before the air conditioning broke.
But they eventually died after.
It was like the first or second week on wild type. And then, man, we're doing better, we're doing great. Our experimental, they look great. And then we came in early, and we're like, whoa, what happened? Wild type were purple and dead. So it was a really quick thing.
Yeah, I mean, we came in, and then a
day they were taking observations on them, they just kept wilting and shrinking and like really quickly.
All right, good work, guys. Congratulations.
All right, guys, while we're setting up, stand up, just kind of shake it off for a second while we're setting up the next presentation here.
very long period of time, which is not very healthy. Also, we would probably increase the sulfur as the amount given. We do not seem to have much effect between a
Crap, it's not broadcasting. No.
It's not always constant as light.
It's broadcasting here.
Not necessarily constant light. Also, various other environmental
I'm not getting.
Once with treatment, and that was 5 grams of Epsom salts per liter. Okay, thanks a lot. Great job, guys.
All right, another stand-up break. Stand up, shake off, take just a minute.
So, we'd like to clarify something that was sort of misexplained from the orange block one presentation. So, the way we applied the salt was actually we bottom watered all the plants.
Right.
And then we did the way we applied the salt waters, we did 32 milliliters that we divided among the 2 pots through like a pipette. It was 7? No, I'm pretty sure.
Oh, no.
No. So I think it was like 32 or 35 of the 6 parts per 1,000 milliliter concentration that you're showing. That part was correct, but the way we applied it was through a pipette over top in addition to the bottom.
Okay, yeah, that's a really important distinction. I'll probably get back to you guys to make sure that I have the details completely right. That's definitely an experiment.
Yeah, I definitely thought it was.
And you know, that's the way it goes. You try something, you never know. Yeah, you know, salt tolerance is really presentation affects that. Thank you guys.
right, back to seat, please. All right, thank you. Our next group up is the Nelson County High School group. Talk about their experiment. So please give them a
My name is Meredith, and this is Bryce Wilson, and we'll be presenting the Nelson County High School Arabidopsis project. Our hypothesis. We predicted that if we plant the mutant and wild type Arabidopsis plants in oil-contaminated soil, then the mutant will do better by being larger and producing more seeds. We believe that the mutants have alternate ways of producing the amino acids, and by planting them in the oil-contaminated soil, they will use the organic compounds in it to produce them.
As a class, we decided to test if the mutant amino acid permease 2,1, also known as AAP2, could improve the growth, function, and appearance of the Arabidopsis. If it is shown that it can improve the growth, function, and appearance of the Arabidopsis, then through further research, it can be found that the Arabidopsis can possibly phytoremediate the oil contaminant in the soil.
Phytoremediation. Phytoremediation is the process of cleaning soil or water by plants that absorb the pollutants in the water or soil. Here we have an example of the John Bunker Sands wetlands in Texas. The river is diverted through a successive area of plants that absorb the pollutants in the water and make it clean enough to be drinkable at the end.
We chose for oil contamination to be our main experimental factor because recently Hurricane Harvey hit strong in the southeastern part of Texas, with the main part of that being Houston. Texas is a huge oil refinery state, and because the hurricane was so bad, oil refineries all across the world were forced to shut down, causing oil leakage to mix into the soils with the already flooded areas from the hurricane. We are trying to see if Arabidopsis can help not only Texas, but the whole world, because oil leakage is a big problem around the whole world. See if Arabidopsis can help improve the environment and save the world. This is the before and after picture of Houston, before Hurricane Harvey hit and after. This is another picture to show how bad the flooding was in the area of the hurricane and show how high the flood level is. Give you another picture to show how bad of damage was done to it. This picture shows that it's not only a big problem in Texas, but all around the southern states in America. It shows that there was 30+ rainfall in southeastern Texas and also Louisiana.
Materials and methods. We top water our plants, our control having only distilled water, and our experimental group having a 15 parts per million solution of the oil mixed with the distilled water. In the next graph, we will have the rosette. Here we see that the mutants followed a fairly similar pattern. The mutant control rising above slightly in the middle and down and falling at the end. While when we compare the wild experimental started small and rose above at the end, while the wild control followed a fairly similar pattern as the mutants, but dropping significantly at the end.
The next slide will show the graph for the goal. As you can see here, the wild control, which is shown by blue, and the wild experimental, which is shown by red, stay at a similar rate of growth, with wild control starting off slightly with a higher vault height than the wild experimental. But later, the wild experimental passes it with a 4.5 millimeter difference at the end. Mutant control versus mutant experimental shows how they stay at a consistent rate of mutant control, staying slightly with a higher vault height than the mutant experimental all the way through, and finishes with a 1.33 millimeter difference. The mutants versus the wild. You can see the mutants did extremely better than the wild types. They show a 76.03 average of difference throughout at the end of this experiment of both mutants and both wild types.
The next graph will show the seed pod count at the end. We see here that when comparing the mutant control did slightly better than the mutant experimental. And when comparing the wild experimental did slightly better than the wild control. But overall, there was a very significant difference between the mutants and the wild. The mutants having more seed pods in the end than the wild. In our analysis, possible sources of error were the light quality, possible insects, and the amount of water that each plant received. And a very unexpected result were that the mutants did much better overall.
In the end, the data collected was very inconclusive. The experimental factor of oil contamination may have been too small at 15 parts per million to make a huge difference in the growth of the Arabidopsis in the end. We suggest that anybody who future researches this experiment will use a more impactful experimental factor than 15 parts per million. Our hypothesis that the mutant would grow larger and produce more seed pods was neither proven correct nor disproven. The Arabidopsis survived, all of them, all four of the Arabidopsis survived, and didn't show any significant differences, mutant versus mutant or wild versus wild, which could lead us to believe that through further research we could find that it could phytoremediate the oil contamination within the soil. I would like to give thank you to Mr. McDowell and the Virginia Tech people, and I would like to give thank you to the BRBGS people as well, and Mr. Sani for allowing us to do this experiment and help us through it.
So, I'm good. Did you guys notice that? So when you added the oil, was the 15 parts per million based off of like what typically happens in an oil spill, or was that just what you guys had found was a safe amount to add? Was that based off anything in particular?
Originally, it was going to be 10 parts per million.
Okay.
But the way we measured it made it so we could do it. It ended up with more oil contaminant in the soil than it originally planned.
And did you guys notice that it built up at all on the soil, or was it go like, I guess when you talked about phytoremediation, did you notice anything happening? Like, could you tell that the oil had been there? Like, was there a difference in appearance or anything?
Not really. There wasn't a very significant difference in the plants. You can kind of tell that obviously it did do some significance, as in the end it had less seed pods and wasn't as tall as the mutant control. But overall, there wasn't like residue of oil on the top of plants or anything, and there wasn't a very much difference. Which is why when we say for future researchers to add a more significant amount of oil.
Yeah, and it's one of those things where I wish we could. I want you to do this again, like right now, and add more, because we don't often tie in. We don't often see groups pick something that's like a big sort of kind of catastrophe like this and tie it in with their research. So I thought that was really neat.
Well, I mean, our class wanted to save the world. So.
Thanks. So I agree that you were amazingly creative in choosing a treatment that's really pertinent and topical right now. That was a good job on that. Okay, so you didn't get a response to the treatment. And, I'm sure this was disappointing, but But, you know, when you say that, that your results were inclusive, I would argue exactly the opposite. I think what your data showed very conclusively was that at the rate that you refine the oil, there is simply no plant response. You know, one of the things that I try and preach constantly to the students in my lab is that there's no such thing as a good result or a bad result if the experiment's done properly. And it's very clear that you did that. And what was really impressive was how closely the measurements that you took of mutant versus control, treated versus untreated, tracked each other. You know, what that tells me is that you did a terrific job of taking care of the plants, and you also did a really careful job of collecting data. So congrats on collecting a really sound and reliable data set that enabled us to learn something about how to do this sort of experiment.
Is that because the mutant did survive in the oil contaminated environment, that they can possibly use, because they're fast growing, they can possibly use to obtain the oil and take the root from it.
Yeah, you're right. That's a good point. Yep. Thank you guys.
All right, guys, who gets the? All right. Good, yeah. All right, please welcome our next presenting group to Madison County High School cohort.
Good morning. My name is Jonah, and these are my partners, Darren, Emily, and Sam. The purpose of our research is to better understand how to harness genes present or absent in Arabidopsis thaliana plants, and how those genes respond when exposed to an environmental factor. This provides the ultimate goal of applying our findings to the improvement of agricultural efficiency.
The modified gene was AAP2, a protein pump responsible for pumping amino acids throughout the plant. As a class, we grew a total of 32 plants, 16 of which were mutants and had the AAP2 gene deactivated through an insertion mutation of tDNA to the protein coding sequence. The other 16 were wild-type plants with a naturally occurring AAP2 gene present. These were divided into two groups of 8, 8 being a control group, and the other 8 being our experimental group that was applied our independent variable potassium nitrate. It is known that the uptake of nitrate through the xylem is an important part of nitrogen metabolism. The forming of organic compounds such as amino acids from inorganic compounds such as nitrate. It is also known that the mineral potassium also serves as a trigger for biochemical and physiological processes known to increase plant growth, yield, and quality. With this, we proposed that we would receive higher plant growth with our independent variable potassium nitrate. Initially, all plants were bottom watered with distilled water every Monday and Thursday for a total of 3 minutes. 22 days after planting, a 2 molar concentration of potassium nitrate was applied. Every Monday and Thursday, observations and watering continued. We measured bolt height and rosette diameter. Rosette diameter was measured using a caliper, and bolt height was measured using a metric ruler. All quantitative data was kept in a spreadsheet, in which we later used to create our graphs, and all qualitative data was kept in a journal along with pictures.
This graph shows the wild-type control versus the mutant control rosette diameters. So this shows only the effect of disabling the AAP2 gene. Growth was consistent until about day 14, which is when the data began to differ. Also notice that drops in the average rosette diameter or bolt height, especially early on, like the ones seen between days 14 and 16, that's due to thinning done by each individual lab. For the majority of the observational period, the mutant plants trended slightly wider than the wild types. They were about 6 millimeters wider at day 24, and this difference slightly decreased to about 5 millimeters by day 31. A similar, more consistent trend can be observed from the graph comparing bolt height. At day 21, the mutants were about 25 millimeters taller in terms of bolt height than the wild types. This difference increased to about an average of 45 millimeters by day 36. These results were surprising because we initially expected the AAP2 mutation to hinder plant growth with fewer amino acids being pumped throughout the plant. However, these results showed the exact opposite conclusion. It's known that the AAP2 gene has an affinity for small amino acids such as alanine, asparagine, and glutamine, but also accepts larger aromatic acids. So we know that the AAP2 gene prefers some amino acids over others. After analyzing these results, we hypothesized that perhaps the AAP2 gene has an affinity for amino acids that do not significantly affect the two measures we recorded, rosette width and bolt height. Further research is needed to determine exactly which amino acids are most responsible for determining rosette width and bolt height of Arabidopsis plants, and also what caused the mutant plants to have a specific advantage over the wild types.
In the graph shown here, the wild type control and wild type experimental rosette diameters are being compared. This graph shows the effects of potassium nitrate. The experimental group rosettes were significantly larger than that of the control. Control and experimental group data remains fairly consistent until around day 21. Potassium nitrate application began on day 22. At the onset of application, the wild type experimental group had around a 4 millimeter advantage over the control. By day 42, it had a 21 millimeter advantage over the control. A similar trend can be observed in bolt height. Noticeable differences in bolt height occurred later and were not visible until around day 28. After this point, the wild type experimentals rose to about taller than that. These results can be supported by our original prediction regarding nitrate and its role in nitrogen metabolism. Therefore, these are the results we expected. Some of the effects caused by nitrate are due to changes in gene expression. It induces for production of genes encoding proteins that redirect carbon metabolism from starch and sugar synthesis to that of organic acid formation. This could draw a link between the amino acid protein pump and carbohydrates that made the experimental group healthier overall.
In this graph, wild experimental and mutant experimental rosette widths are being compared. This shows how the mutant plants responded versus wild type plants to the same experimental genes. The mutant group rosettes grew slightly faster than the wild type plants until day 31, in which the widths were the same and the mutants fell behind them. Growth after day 31 led to the wild type plants having rosette widths of 45 millimeters and the mutants having widths of 40 millimeters by day 42. Overall, we find these results to be within the margin of error. The bolt heights behave slightly differently. The mutant plants surpassed the wild type plants at day 16 and had a sharp incline to 210 millimeters by day 28. Growth after the mutant plants then leveled out and met the wild type heights on day 38, and then which ultimately led to the mutant plants having bolt heights of 10-20 millimeters taller than the wild types. These results were unexpected. We expected the disabling of the AAP2 gene to hinder growth due to a lack of proteins being pumped throughout the plants, without being expedited by potassium nitrate. However, the opposite occurred in the bolt heights. We applied potassium nitrate via bottom watering through the roots. AEP2 is not responsible for the transport of many nitrogen compounds like potassium nitrate. AEP1, which is responsible for the uptake and distribution of many nitrogen compounds in the plant. This explains why therefore, AEP2 does not adversely affect growth heights. These hypotheses begin to explain why the disabling the AEP2 gene does not adversely affect the plant, because AEP2 has an affinity for basic amino acids that do not profoundly affect the plant. More research is needed to determine why the disabling of the gene had a positive effect, specifically growth heights.
Arabidopsis thaliana is affected by a common pathogen known as HPA, a pathogen that grows mildew to cover the plant. The pathogen takes over the AAP2 protein pump and uses the amino acids pumped by the plant for its own nutrients. When the plant does not have the AAP2 gene, it is less susceptible to the pathogen. Therefore, we have found that the most successful scenario for farmers would be that of the mutant experimental group, with its protection against the pathogen and its exposure to potassium nitrate. We would like to thank Dr. McDowell, Ms. Mack, and the Blue Ridge Regional Governor's School. Are there any questions?
So this is pretty interesting because now we have an opportunity to compare your results to those of Gooch and Block 1. Both groups used 2 molar potassium nitrate, but with very different outcomes, right? It looked like the plants that you guys treated, if anything, thrived under treatment, whereas those that the Gooch group treated kind of keeled over. Do you guys have any idea about potential causes for that difference?
We applied the potassium nitrate bottom watering, which is where we think because the AEP1 said that it is responsible for the uptake and distribution of all the nitrogen compounds. So the AEP1 is located in roots where we apply the water. That's nitrate. I think they applied it through pipettes. I think that was.
You did bottom water, or?
We injected it right.
Really? That's potentially a big difference, isn't it?
How many times a week did you treat with potassium nitrate?
Every Monday. See, we only did it every Friday. We only did it every Friday.
When you were bottom feeding, how much of the solution did you add in there?
Okay, it was a 2 molar concentration with 1.5 grams per 1 liter water, and left it soak for 3 minutes, and then they were taken out and transferred on to fire.
What's y'all's interpretation of this?
Well,
the plants would have taken up less solution because it was added in the water. We added 1.5 milliliters of our solution on top of each of the plants. So I think that's a big difference.
therefore, by them adding the water, we'll add the solution to the water and mixing it directly, and we will use the solution.
We went back and forth in our class, and we got back to the solution of were we going to put it on top or were we going to do it through the roots, and we decided to do it on top.
So that's how fertilizer would be.
Yeah, that was fertilizer would be added to the plants.
Yeah.
It was a lot of water fertilizer.
What are y'all's thoughts about this? And I should say that, for these experiments, there's no right way to do it, no wrong way to do it.
I think a big take-home message all of you should be listening to right now is that's one of the big things we focus on in science experimental design. And this is a case in point right here that you change experimental design and you had huge differences. So it is very important for your designing experiments on how you set it up.
Yeah, that's exactly why I'm drawing this discussion out. This happens all the time in science. Different groups do what seem to be the same results, but with different outcomes. And then the challenge is to get together and think about, what the causal differences are. And that's often a path that leads to some really important new insights.
Our first application of the
And what you guys did very, very well there was, you know, you were basically alert. You had situational awareness, right? You saw that something was happening that didn't look right with the treatment, and so you adjusted the design of the experiment. You know, that's what a good experiment is to do.
So after the third time y'all put the solution on top, it seemed to be doing damage to the plants. But while we were doing that, there was a week or two that it helped. Question for you, why did that happen?
I think the reason that that could have happened is not over application, but a lot of it on the leaves might have started hurting it due to nitrate. Nitrate has some properties that can damage a plant without potassium. That's one of the reasons potassium nitrate. And if you have it going to roots, then there's a less of a possibility that. And since it stayed on there after you guys applied it, and when we applied it, it was applied and then taken out of the solution and then put back in the normal environment.
So like when you guys are spraying yours on there, we also saw the effect that after we were adding our nitrogen solution in there, it was growing better, but some of the drops from the pipette actually got on the leaves. You can see those leaves starting to deteriorate, so that's probably why we're seeing that horrible effect in the beginning.
All right. This is a great discussion. Well done.
All right, ladies and gentlemen, we have come to that favorite part of a field trip where so, hey guys, need a little attention here so we can do this smoothly because we have lots of people here. There are tons of pizzas out there, so nobody's going to go hungry. But what we have to do is a couple of things. First of all, we're going to have, we are going to, at the end of the lunch period, we're going to have a little bit of time for a little bit more poster session. I don't think Dr. McDowell got to see everybody, and I don't know if all the poster groups got around as much as they needed to. So we're going to use a little bit of time in about 20 minutes to do that. So what I'm going to do is have the poster groups go out and get pizza second. They're going second because the adults are first. So adults, you guys can head on out and grab some pizza, and then I'm gonna start calling up poster groups by schools. So we'll do that. Hey guys, couple things. As you know, you kind of have the whole room, this whole place for eating, but we're going to ask you not to move the chairs a whole lot, just because we're going to need to be like this for several more presentations. So big circles and stuff like that, we're going to have to rearrange. So keep the chairs in the rows and stuff. All right, next up. When I call your school, this is for the poster group only for right now. Okay? So when I call your school poster group, head out that door, pick up pizza, come on back in to those doors down there. So, all right, poster group, green. No, this is poster presentation group. Poster question group are going to be next, actually. Poster group, Goopson 1. Poster group OCHS 1. Poster group Fluvanna 4.
Poster group Nelson. Poster group Madison. Commissioner Garza. Commissioner Precinct 4. First group, Orange 4. And first group, Savannah 2.
Catch on that.
Okay, next one we're going to do is get the people who are posted, the questioners for the posters, because you guys will enjoy it. So, we're going to work back the other way. Questioners for Fluvanna 2. Don't worry, there's plenty of pizza out there.
All right. Yeah, I'm gonna. Well, okay. I'm doing all the poster folks first, so we get them, and then I'm gonna do the presenters in that order. Yes.
OCHS 4. Questioners. Goochland 4. Questioners.
We're best to go last.
Louisa questioners. Madison questioners. That's the question, folks.
We have questions.
Okay.
Nelson questioners. Savannah 4 questioners. Orange 1 questioners. And Goochland 1 questioners. All right, green questioners.
Mercy.
All right, next up, we're going to do the presentation groups. We're going to do these in order. The ones who still have to do their presentations afterwards are going to go first, so they have more time to do
Oh, guess what? We're going home.
So, Louisa, presenters. Groups on the floor, presenters. Orange school presenters. Blue code 2 presenters. Green presenters. Green presenters, indeed. Group 1 presenters. You're welcome. Orange 1 presenters. Going twice. Three times. Orange 1 presenters. Savannah 4 presenters.
Guess what?
Nelson presenters. And Madison presenters. Everybody that has done that pizza yet.
And then who else we left out? I'm always wondering what's coming out. Push this back just a hair. Yeah.
We're definitely not going to need all of that.
Yeah.
We're doing it a little bit right now.
Right.
So I think that sounds good.
Yeah.
That was awesome.
Yeah, some of that is right. just wrecking the place. All right.
I definitely think so. I think that we would just want to maybe pick two extremes as one variable, like something like that. I definitely think.
Yeah.
And that would not affect any of our normal day-to-day what we do as far as targets. We will affect the very, very end. We'll do that. I think that would be great.
Okay. Something to think about.
Like right now, it's 11. Yeah. You definitely are. Right. So I'm like, I'm happy to add anything like that to our deal. I think so too. Yep. I think that'd be fine. And that would, if you're doing a collaborative thing too, that would not be a bad time, depending on who's on that trip. Just check in. I don't know how
What's your problem?
How are you doing, dude?
Boom!
It's not.
Great house, just one front door. What? They said you were wearing a sweater and everything. I'm sorry. No, it's the great house. Well, if I forget that, it's all
No! No! Stop. I'm stealing my line. I'm not having the word for word. Look, I'm giving my line. I'm going to say it. I just look loyal to my back. That organ is better as shown, no!
In the middle of each presentation, oh, that was
Ooh, that was feeling nice.
You keep saying that. You're out on
I'm gonna screw up big time. Don't screw up. Well, then maybe you should try out for drum bass. That's what you have to do. We're just gonna look over and Jimmy's gonna be like, I was doing like
Dylan, what great practicing of your speech. We were. So you use your
of course.
So worried I'm gonna make you guys mad. I'm worried I'm gonna make you guys mad.
Alexis, should we go over there and just wait for the call to end?
I think I got it, but I'm worried.
Should we go over there and wait for the call to end?
You should do high five. Hit it.
No. Get ready. No. Everybody's hand like this.
What? I'm so
Ready? Set.
Okay, you gotta do it again now.
You're gonna get
All right, Louisa Peeps, y'all ready?
All right, ladies and gentlemen.
There are Louisa group. Who's presenting? Thank you. We're going to get underway with our final presentations of the afternoon. Some people have already done it, but please give the Louisa group a warm welcome.
You hear me? My name's Dylan C.
I'm Alex Legon.
And I'm Ethan Snyder. We're from Louisa County High School, and our experiment tested the effects of magnesium sulfate on the Rabbitops Valley Island. Magnesium sulfate is an additive that, when given to plants, has been shown to increase their growth and reproductive rates. We gave 0.3 grams of magnesium sulfate mixed into 40 milliliters of distilled water to our experimental groups twice a week on Mondays and Fridays throughout the duration of our experiment. We hypothesized that when given magnesium sulfate, our mutant experimental groups, the disabled AT2 gene, would exhibit an increase in rosette width, bolt height, and seed reproduction.
This graph shows the exponential growth of rosette widths and Arabidopsis. Around 35 millimeters was the average at the top of their life cycle, and 2 days after hitting this 35 millimeters, they began to decline downward. This graph shows the meat experimental plants and all the others quickly exceeded their life cycle, as an average Arabidopsis life cycle is 6 to 7 weeks long. The meat experimental plants also have the largest rosette widths, have the highest folate counts, and reach reproductive maturity faster than any other plant. This graph shows a comparison of the wild-type control and mutant control Arabidopsis. This shows that the ACT2 gene actually does have an effect in the growth rate of plants, as the mutant control started off higher than the wild type, and the wild type control, with the ACT2 gene still enabled, ended up having the highest result. This graph shows a comparison between wild type experimental and mutant experimental. This shows that the wild type experimental and mutant experimental were both actually affected by magnesium sulfate. This stressor did a better job at being effective. This stressor was affected the magnesium. This stressor affected the mutant experimental more in a more positive way than the wild type experimental.
Graph 4 gives a comparative glance of all of the bolt heights on all of our Arabidopsis in our experiment. As shown, the mutant plants, specifically, the control groups, specifically the mutant control followed by the wild type control, grew taller bolts. This suggests that magnesium sulfate has a negative effect on the growth of Arabidopsis, and that when the ACT2 gene is disabled, that the plants want to reach maturity faster, can produce more siliques, and that way they are not focusing as much on the height of the bolts. Graph 5 compares the control plants, and as shown, the mutant control is taller than the wild type control, but they both even out about even out at the end, though there's not much difference in these plants to be of much significance. As shown in graph 6 with the experimentals, there is a much larger difference between the mutant experimental, which grew taller, and the wild type experimental, which was shorter, which suggests that with the ACT2 gene disabled, the plants can respond better to the stress of magnesium sulfate.
This graph shows us data concerning the silique reductions from all 4 of our observed groups. As you can see, the mutant experimental group came out with the highest amount of silique reduction, whereas the lowest amount of silique reduction exhibited was the wild type experimental group. It's also worth noting that all data collected concerning silique reduction from group 2 was excluded from the class data due to errors that will be discussed later. This graph gives us a comparative glance at our 2 control groups. For the majority of the experiments, the mutant control group was exhibiting a higher amount of silique reduction. However, in the end, the wild type control group did surpass the mutant control group with higher silique reduction. In this graph, it is very clear that the mutant experimental group exhibited far more silique reduction than the wild type experimental group. And this leads us to conclude that the wild type experimental group did not respond well to magnesium.
This image shows the purple leaves that were an effect of anthocyanin being present in the rosette. This may have been a result of overwatering. This is an unknown white fibrous growth that we found on our wild-type experimental plants in group 3. This may have been a result of humidity. There was also many places where there may have been error in the experiment. Plants, when they were growing, began to lean outside of the pot due to withering at the base of the bolt and rosette. This withering caused them to tangle together and had to be touched to be measured, which caused the siliques to be fragile and begin to fall off. Group 2, as mentioned earlier, was excluded from the data collection pool because they had an error in counting their silique pods. Calipers were used in some groups, but some groups also used
regular rulers, and they both have different margins of error in measuring bolt height. Therefore, the bolt height may have had spikes or differences in error.
In conclusion, we said that the hypothesis was not supported or it was rejected, though there was partial supporting when, because the hypothesis stated that the APT2 gene or the Arabidopsis with the APT2 gene disabled that is watered with a solution of magnesium sulfate and distilled water would develop wider rosettes, taller bolts, and produce more saliques. However, although more saliques were produced and the rosettes were wider for the mutant experimental, not as many bolts were produced. Bolts were not taller. However, the growth rate was increased throughout the lifespan of the experimentals, and you can see that in these pictures up here. For over about a 3-week lifespan, or just over 3 weeks, the plants went from the early stages of their life cycle in rosette development to almost at the end of their life cycle, with most of their saliques already being produced. However, evidence for why the bolts were shorter, but why more saliques were produced, was stated earlier when the experimental plants were trying to reach their reproductive phase sooner, so that they could produce more saliques in response to the stressor. This means that overall we do not know the effect of magnesium sulfate completely on Arabidopsis. We also don't know why in our data the experimental plants or the control groups grew better, but why the control groups or the wild type grew better. We would like to thank Dr. McDowell and Virginia Tech, Blue Ridge Virtual Governor School, and Mr. Caraway for the amazing opportunity to perform this experiment. Does anybody have any questions?
During your research, were you able to find the average rate for salique production with Arabidopsis plant?
On average, the plants produce about 11 saliques at one time.
Thanks, great job. So we have another opportunity for comparison because William Monroe tested copper sulfate, and I asked them a question about whether they thought it was the copper or the sulfate that was causing the effects. But before we get into that, I'm also curious to hear about calipers versus rulers. You guys use both. Which do you think is easier and which more accurate?
So we thought that when measuring the rosettes, at least I did, calipers were a lot easier, just because it was one quicker for our class period, but it also provided a little bit more accurate measurements. And then for when we measured bolts, at the beginning calipers were easier because also it gave more accurate measurements. Then at the end, the bolts just were too tall for the calipers to be used. So we ended up using rulers, and that made it a lot easier. I'll probably switch back and forth.
Yeah, that makes sense. Okay, so William Monroe, what did you guys think about, where is William Monroe? What did you guys think about their results?
I think some of the results were similar. They had the experimental plants had significantly a smaller bolt height, but more seed pod production. Is that right? Than the control plants. And so our control plants actually had a higher seed pod count. However, I think the seed pods are subject to a lot of error because they can be very easily knocked off. And the bolt height really interests me because that's a similar result. Our controls had smaller bolt height, and yours controls had smaller bolt height. So I would say that probably the sulfate is affecting that. Did you guys notice any discoloration in your leaves at all, in terms of yellowing?
With the withered around the edges of the oak.
Okay, so that's potentially a cause of the sulfate. Did you have any light discrepancies in your experimental design? As in, like, were there any inconsistencies in the light your plant received?
There weren't new windows. They were all the windows here. Both windows were shaped, but I didn't want to take all the light out, but they were off in a corner away from the light. And they had a light that was on counter that was put over top of them. And then just plants, experimental plants that separate from.
And so I think based on, you guys had very little experimental error in your lighting. And so I think based on that and based on our results, we can conclude that the sulfur and the sulfate compounds was responsible for the yellowing of the leaves in the sundial light.
That's what I take away from this. And as I mentioned in my kickoff talk, we're very interested in also in sulfur metabolism in our labs. And the experiments that we've done so far have involved withdrawing the sulfur using a hydroponic medium. But I think it's probably worthwhile for us to go back and do some experiments where we just add sulfate to soil and do pathogen experiments on that. So thanks, guys.
All right, ladies and gentlemen, you're hitting the home stretch here. Please give these last few groups the same attention you've given the rest of the group throughout the day. So I'm going to turn it over to the students from Block 4 team. Give them a big hand.
Hi, I'm Bryden Roberts, Ethan Wenzel, and Stuart Taylor, and we are representing Houston Block 4. So, for our variable weeds, magnesium sulfate tested the effects of that on our Arabidopsis plant. We had two genetic variations of the plant, one that contained a gene that is naturally found in Arabidopsis thaliana and one that did not. And we tested our experiment over the course of 5 weeks. And before we conducted our experiment, we formed a hypothesis, which was if magnesium sulfate is added to the plant soil, then the wild type plant will become more stressed than the mutant due to the sulfur.
So across the world currently, there are hundreds to thousands of harmful pesticides being used in farms across the globe. Now, all of these pesticides either harm the plant, harm the environment that they're in, or harm the society around them. Whether it be killing the plant, making the soil poisonous for future crop yields, or just making the soil inhabitable to other plants, or the society by these pesticides are harming the people who eat these plants. Now, we took away from this that we're trying to find a sort of semi-natural pesticide by testing these things, testing the magnesium sulfate and the AAP2 gene. And we're trying to see how these things affect the plant so that we may better understand how these are variables that we've all been testing affect the plant.
So a little bit about Arabidopsis. This is known as a model organism for scientists because it has a very short lifespan. It is a very small growing plant. It has no economic value, and it's found pretty much around the world. It has a very condensed population in Asia, but it can be found everywhere else as well. So Arabidopsis contains this gene called the amino acid permease 2 gene, or the ACT2 gene. And this gene produces a protein that moves amino acids out of the cells, and this can be found in the phloem of the cell. And this gene was disabled in 2 of the plants that we tested.
This is the equation we use to find out how much magnesium sulfate we should add to the water. We ended up using 492 grams per liter. And we would take a pipette and apply the solution via pipette, 10 drops per plant. The plant received 16 hours of sunlight and was watered twice a week, and we applied the solution once a week.
So when we started our experiment, we planted our plants, and then 2 days later, we came to see Dr. McDowell's presentation here for the kickoff, and we formulated what variable we were going to use here then, and then went back to our lab to create the variable to add to our plants. At the beginning, so to start off, how we separated these two things was we took our experimental and our control into two separate containers for watering, just in case there was no cross-contamination, and so we wouldn't have that error as a possibility. We watered on Tuesdays in about a 5 centimeter deep water bucket for about 15 minutes for all of the plants, and then we added our solution directly to the soil, not onto the plants, via 10 droplets out of a pipette from our solution. As the weeks continued, and we continued to do this on Tuesdays and then add the solution on Fridays, we noticed that our experimental became under stress very quickly and bolted before any of the other plants, leading us to believe that our variable that we added to the soil caused these plants to be under stress, leading to an earlier bolt to get their seed production undergoing so they can reproduce before their life cycle gets cut off. We also noticed by the end, there was no distinct difference between the two plants and how they looked, our control and our experimental, because we believe that just because we added our variable, it speeded up the growth, but by the end, they both were very similar, just our control had grown a bit slower during the process of this entire experiment. With this, we continue to notice that the bolts of the experimental grew exponentially quicker than the control. By the first 3 weeks in, the bolts had grown to their full length, and it continued small increments from there, but nothing that visible was seen from there on out. The measurements were pretty similar, and for the control, they grew at a slow and steady pace that we were expecting, and all the dates that we were expecting the bolts to show up and the seed pods to show up were accurate.
This is a graph of the rosette sizes of each of the plants. If you can't see the lettering because it's small, the blue line is the average mutant control. The red line is the wild type control. The yellow line is the mutant experimental. And the green line is the wild type experimental. And each of this is measured in centimeters. As you can see, the mutants were largely had bigger rosettes throughout the experiment than the wild type, which shows better growth and less stress. The blue line is the wild type control, the red line is the mutant control, the yellow line is the wild type experimental, the green line is the mutant experimental, and this is the average bolt height. The wild type controls were bolted slower, which shows less stress, as early bolting is a sign of stress. And this is a graph of the number of seed pods. The blue line is mutant control, the red line is wild type control, the yellow line is mutant experimental, and the green line is wild type experimental. Both mutants had far more seed pods earlier than the wild types did.
So at the end of our experiment, we concluded that the magnesium sulfate actually stressed out our wild type experimental plant, causing it to have a rapid bolt height growth, rapid rosette diameter growth, and rapid increase of seed pods because the plant wanted to reproduce faster in case it had growth.
The AAP2 gene did not have much effect on each of the plants, which showed that plants without the AAP2 gene can function properly in sulfur-rich environments.
To conclude all of this, we put all of our data together, and we realized there was less of a difference between our experimental and our control from our wild type and our mutant. There was more of a difference there. So we did realize that the AAP2 gene, while not that that impactful had a small effect on the R variable put into the solution, put into the soil for the plants to absorb. From this, we as a class concluded that at the end of this, we had way more questions than answers. And a lot of us were very upset about that. So we sat back and examined how this process was, and we realized it was the process that was the important part of this experiment. It wasn't the outcome. It was the going through all of the classes. Every single class in here could take a different path down this experiment. You could have done different variables, treated each plant differently, different days, and different environmental variables for all of this. So the way the process went was the important part we felt to this entire experiment. It wasn't what we got out of it right at the end. It wasn't the answers.
And what we would do, we concluded from our experiment that to redo this experiment, if we were to start over, we would reduce the amount of magnesium sulfate that we used, because we did use a super high concentration of that in our solution, that we would just decrease that to see the effect that that has on the plant as well. And one last thing, our class, there is an amazing person who came up with a quote that really sums up this entire experiment all together. My dear friend Claire Anderson came up with a quote that I've got 99 problems and Arabidopsis is all of them.
Any questions?
So I've got a couple of comments. Yeah, 99 problems. It's similar for me, but usually administrators are responsible for about half, and the plants are responsible for the other half. I do completely agree that that it's the process that's important. That's something that I almost constantly have to remind my students of, because they're they're results focused, as they should be. But it's really all about the training. I think that's particularly true in this context. So I think that's a really good insight that you guys can do. I'm also pleased that that you had more questions than answers at the end of the experiment, because again, that's how science works. That's good, because it keeps guys like me in the job. Now, again, we have an opportunity for comparison. So I'd like to come back to Louisa and sorry, my computer froze up, as did my brain, and William Monroe. So I think we have a pretty clear consensus that's emerged from these three sets of experiments, right?
Yes.
And that's a good thing. That illustrates another important aspect of how science is done. As you guys have clearly developed a sense of, even in the most controlled experimental setup, there are often small variables that are just impossible to control. The AC goes out. Okay, the only place that you can put your growth racks is next to a window. So you get some bleed in from that. It's just that way in university level research labs as well, even the best resource ones. So really the gold standard for expecting, for assessing the validity of some experimental observation is whether it's reproducible or not in between different groups in different places. And I think that's what we have here. Great job, guys.
Home stretch. All right, who's got what? All right.
My name is Daisy. This is Jenny. This is Rory, and this is Eva. So we conducted an experiment to determine the effects of disabling the gene AAP2 gene on Arabidopsis thaliana plants. And the AAP2 gene is an amino acid protein pump that helps to distribute the nutrients throughout the plant. A disease called downy mildew takes advantage of this protein pump by stealing the nutrients before they can be distributed throughout the plant. So we chose the amino acid lysine as our variable, and we chose it because it's known to improve crop yield and overall health of plants in agriculture, and it also helps to amplify plants' defense against pathogens. So knowing this, we created our hypotheses, and these are that the wild type experimental was expected to grow the best because it has additional nutrients added, and the wild type control was expected to provide a baseline to the other plants because it does have the AAP2 gene, so we're seeing the effects that vary. The mutant experimental was expected that adding lysine may counteract the negative effects of the AAP2 being disabled because of the additional nutrients. It was expected to have the worst growth overall because it has no amino acid pump to distribute the nutrients throughout the plant. So we applied the lysine with a 40 milliliter solution with a 15 millimolar concentration of lysine. And to collect the data, we measured the rosette width and bolt height, and to measure productive success, we collected seed pods.
So this is our average rosette width graph. In the beginning, all of our rosette widths were just about the same, but around day 25, they started to vary a lot. So both the wild type plants seem to follow the same trend and stay pretty close together the whole way until the end where they split off. The mutant control rose above the others in the width, but it started to level off around day 28, while the mutant experimental didn't grow much at all, and Rory will tell you why.
There were several unexpected results in the data, and one of them was that the mutant control group grew much larger rosettes than the rest of the groups until the end where it dropped off there. And this is pretty unexpected because theoretically the mutant should be much lower than the rest because an amino acid pump is removed. And another one was that the mutant experimental grew much worse than the mutant control, even though the wild types remained fairly even. And this is most likely due to the fact that the amino acid pump was removed, and so the plant couldn't recover from the lysine addition as well as the wild type could. And also, the experimentals had a much higher rate of growth at the end than the control groups did. And this is very similar to the fact that they're much more green and lively at the end than the control groups at the top. And this is very likely due to the fact that the lysine increased growth and life expectancy. And this is pretty unexpected because originally they turned really yellow, and we thought they were gonna die. So we were basically seeing how well they could recover, and they ended up coming out like a lot better. And the reason that the initial growth was so much worse than the was because the lysine concentration was probably too high, and eventually they recovered after the application stopped, and they were able to utilize the nutrients.
So this is our average bolt height graph. As you can see, the mutant control seemed to follow the same trend as it did before, rising above the others, except this time it stayed the tallest out of all the bolts. And the other three varied differently throughout the experiment, but all ended around the same area with no significant difference between them. A visual representation of this is you can see the mutant control was obviously the tallest in its bolt height, while the mutant experimental seem to lag behind. And you can switch the control groups and the experimental groups. The control groups straight up with tiny leaves on them, while the experimental have nice thick leaves and seem to wind and twist around each other.
Now, again, the fact that the mutant control is so much higher than the rest of them was fairly unexpected, but less so for the bolts, because initially, when it's under stress, it grows bolts much faster than the rest, so it can reproduce faster. And the the experimental plants had much more average growth, and that was pretty unexpected because originally we thought they would grow the best, and after the initial lysine addition, they seemed to be dying, so we expected them to grow the worst. And in the end, the fact that they were in the average region of the graph showed that it contradicted both the original assumptions. And we're unsure why the plants grew, the experimental plants grew so much more windy than the control. They were a lot more green and lively at the end of the growth period in the control group, and this was a hypothesis since lysine is a nutrient, even though the lysine appeared to be hurting the plant originally.
So this is our average number of seed pod graph. There was no significant difference between the plants. But they all varied from 2 to 3 different number of seed pods.
So as Jenny said, there was very little actual variation in seed pod quantities, which was odd, as previous research has suggested that mutant control would have more seed pods than the rest. Now, while there is very little variation, you can see clearly that both of the experimental groups had more seeds than either of the control groups. And then here's a visual representation of the seed pods. There's clearly no difference in coloration or size. So overall, the wild-type plants grew more efficiently than the mutant plants, as is most efficiently displayed, I believe, by the bull-type graph. Both of the mutant plants began to bolt far before the wild-type plants, showing a possible decrease in life expectancy. And in the beginning of the experiment, lysine seemed to hurt the experimental plants. However, midway through the experiment, they showed a remarkable comeback, possibly meaning that in smaller doses, the lysine would make a difference in their growth. I believe this is best represented by this picture over here. On the left, you can see the mutant experimental, and on the right, you can see the mutant control 44 days after they were planted. Now, the mutant control on the right is clearly a dark brownish color and appears to be nearing the end of its days, while the mutant experimental on the left has a clear, vibrant green color and is growing perfectly fine. Most of the data, unfortunately, not support our hypothesis of lysine counteracting the downsides of removing the AP2 gene. However, we believe that in smaller doses, the lysine would assist in the growth of these plants. I'd like to thank Dr. McDowell, Virginia Tech, and the BLVGS Association for allowing us to do this project.
Great job, guys. Can you go back a couple of slides to the conclusions? Because I wanted everyone to see this and marvel at it. Boy, what a difference a little lysine makes. Now, how do the plants look when you're comparing the wild type control and experimental? Do you see the same thing, or is this something that's specific to APT2?
Very similar. The wild type, it was greener than the mutant control is, the wild type control, but other than that, it was pretty much the same colors.
Oh, right. Excellent. Okay. So one of those is not like the other ones. Go through each one and tell me which one is which. Mm-hmm. So the mutant controls the oddball, isn't it? What do you make of that? It's okay to be bewildered, because I kind of am too. Yeah.
Evidently, the lysine wasn't great for either of the control plants, both for smaller and for the
Remind me how you did the lysine treatment? Okay, so, and how many different lysine treatments did you do over the course of it?
We actually did two, because once they started dying, we stopped the treatment.
Oh, yeah, right. Yeah. But then they bounced back and did a lot better. I think your interpretation was good. It was probably too much at first, and then as it became diluted and metabolized, it was probably. All right, good job with that.
Let's go! Let's go!
Apparently they have a fan base. Happens all the time in scientific presentations, doesn't it? All right, ladies and gentlemen. We've arrived at our final presentation of the afternoon. Please give me a warm welcome.
Hello, so today we are presenting the effects of acid rain on Arabidopsis thaliana. I'm Claire Kent.
I'm Mackenzie Moore.
I'm Maggie Woods.
And I'm Alex Bofi.
Our hypothesis stated that if a pH downer was applied to water, then the wild-type experimental plants would show more growth than the mutant-type experimental plants. We decided to test this because acid rain is a relatively common occurrence in many parts of the world due to widespread industrialization. Typically, acid rain has a very negative effect on the health of plants, and since the mutant-type plants are missing the AAP2 gene, we predicted that they would react in a more negative manner than the wild-type plants. For our variable, we applied an acid rain simulant to our plants due to this being an increasing environmental issue as our world continues to industrialize. Acid rain is said to negatively affect plants because it dissolves the important nutrients and minerals that are supposed to enter the plant to help plant growth. To simulate this acid rain, we applied pH downer on Wednesdays during our experiment with watering regularly on Mondays and Fridays. The pH downer was bottom watered into the plant at a 1 to 25 ratio of distilled water for a 2-minute period each treatment. The pH downer consists of citric acid monohydrate, and the citric acid lowers the pH of the soil to a 2.0, which could possibly become toxic for the plant due to the lack of movement of minerals into the plant. The pH was lowered as the soil absorbed the citric acid, which it then entered the plant and hindered its growth.
The average measurements of rosette width of all Arabidopsis plants were measured around a 6-week period of time. During this time, the acid rain imitation didn't have much of an effect on the rosette width other than the gap that you can see which occurred during week 4. We believe this gap occurred due to the lack of nutrients in the soil. However, during week 5 or 6, the experimental plants bounced back to, the wild type experimental plants bounced back to the growth of the control plant.
Alrighty, result differences in plants. So we're going to start off by talking about the different overall size and actually the effect of our pH downer. And in order to do this, we're going to go back to the graph on the previous slide.
So you can see in the blue and green lines, we have our control plants, and just below that in our red and yellow lines, we have our experimental plants. Now at the very end of the graph, it represents actually the end of the 6-week growth cycle of all of the Arabidopsis plants. Now, at the very end of this, it actually represents a 3 millimeter gap on average within all of our plants, showing how, based on the graph data, there actually was no correlation between the AAP2 gene and actually our variable. As you can see that wild and mutant plants actually had no difference within their two groups of controls and experimentals. However, this does disprove our original hypothesis that wild-type plants actually would grow more than the mutant types. But it does prove other predictions that certain plants would grow less than others. Now this brings me to my second point. We need to talk about the different colorations that we noticed in plants. Now in the 5th week of the 6-week growth cycle, we started to notice that all control plants actually turned a purple color, while the experimentals still had the normal green color. And this is most likely due to a change in pigment. Now pigment such as chlorophyll, we all know makes plants green. However, there's two other main pigments that we probably don't know about, including xanthophyll, which have to do with the coloration of red, yellow, and orange, and the one found in this plant called anthocyanin. Anthocyanin has to do with blue, indigo, and violet colors. Now, generally, it's going to switch pigments because it has to do with a lot of stress being experienced on the plant. And this stress can come from a variety of variables, but it's most likely because of the reproductive cycle that it was going through in the 5th and 6th weeks. And we can see that it changed the pigment color so that way it could appeal to a larger light spectrum, so that way it could get more nutrients from the same amount of light in a smaller amount of time.
This is our graph as our average measurements were taken for bolt height over the 6-week period of the experiment. As earlier stated, the rosette widths for the wild type plants were greater than the mutants, but the mutants actually bolted first during the 6th and final week of our experiment. As you can see, our mutant control plant was the most consistent and ended the experimental period with the most well-developed bolts, while the wild-type experimental was also very consistent in the fact that it had no red growth at all. And the wild-type control was consistent until it grew 11 millimeters within 3 days between October 27th and the 30th. All of this brought us to the conclusion that the mutant control was the most successful at reproducing because it ended the experiment with the tallest bolts that were most capable of reproducing, while our wild-type experimentals were least successful in reproduction because they ended our 6-week experimental period with no bolt growth.
The effect of our variable on our bolt height. Now, as I'm sure all of you already know, the bolts represent the reproductive mechanism inside the Arabidopsis plant. Now, what happens is it creates seeds that are then dispersed onto the ground so that our Arabidopsis plant can have offspring and grow again. Now, if you look at the picture over here, we have our wild control, our mutant control, our wild experimental, and our mutant experimental. Now, you can notice that only 2 out of 4 of those plants actually grew the bolts, and those are both the control plants, while the experimentals simply did not. Now, this is most likely because of our pH downer, which was our variable. What happened was the variable actually got rid of nutrients that the experimental plants needed to grow these bolts, and instead of trying to grow them at all, it simply forgot about the reproductive step, since it knew it would not be able to grow them. However, you can see the opposite happened in the controls that had the amount of nutrients that it needed to actually grow the bolt and start a reproductive cycle. Now, this can once again be related to actually the strange coloration that I talked about earlier. We can see with the bolt heights that actually grew in the control plants, that was the stress that I was talking about earlier that caused it to switch to a different pigment. It switched to a different pigment so that way it could appeal to a larger light spectrum to get the amount of nutrients it needed to start the reproductive cycle.
In order to keep the possible percent error of our experiment low, we made sure to take precautions while potting the soil. While potting the soil, we wore gloves and made sure to not directly breathe on the plants. While also doing the experiment, we made sure to keep only one independent variable. By doing this, we made sure to rotate the tray every Wednesday to keep an equal amount of light on each plant. Now, while every experiment has some errors, ours did as well. Most of the pots grew either mold or algae. We believe this affected the color of the leaves and stunted the growth of them. However, in future experiments, we probably should have used 2 trays instead of 1. Considering that our experimental plants were treated on Wednesdays, they could have had some excess treatment on the bottom of the pot. Then when putting them back into the tray with the control plant, if any of that excess treatment could have leaked into the tray, the control plants could have accidentally soaked that up through the roots, causing a contamination.
The results of this experiment are significant because it demonstrates that a plant with a missing AAP2 gene can survive just as well in an acidic environment as a plant that has the AAP2 gene. Now, both of the experimental groups displayed a delay in bolt growth, and they both produced more chlorophyll than anthocyanin. This means that by planting mutant crops, that could present a viable substitution for using harmful pesticides to prevent pathogens such as downy mildew. Now, for a more specific example, crops such as rice or soybeans that are susceptible to downy mildew could possibly be replaced by such a mutant variation that has the potential to survive just as well in acidic conditions. Right. Thank you for your time. Do you have any questions?
So for sure, the effect of the treatment was the most surprising result that I've seen out of all of these. I'm sure it was for you too. Do you think it's possible that the change in pH liberated some nutrient in the soil that would be otherwise limited? I mean, that's a soil science question, I guess. I don't know anything about that. I've got a question for basically for everyone. So at the end of their experiment, and I think I've noticed this across classrooms, by the time the plants had bolted and the experiment was pretty much ended, the control plants generally are purple. They're stressed out with anthocyanin. Is that kind of a general truth? I see a lot of heads nodding. Did anyone else see plants at the end of the experiment that looked as good as the ones that they treated with pH down at week 8 or whatever? Okay, so from that I interpret that under our collective experimental conditions, the plants are getting stressed out, and it may be that your experiment has revealed that the source of the stress could be some kind of limiting. That's my take on this. I don't know. It's pretty cool. What do you think would have happened if you'd applied the pH downer directly to the plants with a sprinkler or something like that? Yeah, that probably would have gotten ugly pretty fast.
Considering the fact that acid rain, when it precipitates on the trees and plants and also in the soil, do you think that if you had applied this from like a rain effect, it should have received greater drastic results and roughly similar results? Yeah.
There probably would have been more structural damage to the plants as well. But we wanted to test what happened when the plant actually sits in it instead of what happened structurally.
Had much of a difference if you did it all up. You put all the experimental treatment at once and then measured it over the course of the next.
So like, if we did one treatment over instead of doing one treatment.
Yes, like as if you were simulating actual acid rain, because you would pump down all at once, and then there would be a period of no water, no acid rain.
to like only the one treatment. You know what I mean? If we only would have done one treatment, then it wouldn't have been as big of a stress factor as doing it every month.
You're saying that all the solutions that you use throughout the entire process, instead of putting it periodically, do the plants every week, he was saying that what would happen if you just added all of that solution the first time? Like just like when it rains, it doesn't rain every day or every week necessarily. What would happen if you just did one treatment and then reported the results over the next week?
Basically an acid deluge.
Yeah.
We wanted to test it like to see if stress was needed for mycosynthesis, and so the concentration we had was just enough, like challenge it a little bit without actually.
And that's a good design. I think you also did a great job of picking a tree.
All right, ladies and gentlemen. Yes, you are done. Give yourselves a hand. Nicely done. I think we also owe a big thanks to Dr. McDowell for taking a lot of time, effort. As a token of our appreciation, you get your own custom Blue Ridge hat. You take back with you. There you go. Very nice. Excellent. Thank you. All right. Any final words to this rowdy bunch of fifth graders?
So honestly, this was inspiring. I really enjoyed talking with you guys and seeing your results and all your hard work. Yeah, I learned a lot too. So if you'll remember, you know, the dream that I kind of described in the kickoff presentation was to eventually use Apt2 as a tool for disease control. As the Fulvana group ended up their talk, which is a perfect segue, you know, what I'd like to be able to do is create Apt2 mutations in crops like soybean and use that for disease resistance. Of course, my fear in this is that the Apt2 mutation would have negative side effects on the plant because after all, it's an amino acid pump that presumably translocates amino acids to the places in the plant where they're needed for growth. So you'd think this kind of mutation would be either bad for normal growth or bad for stress responses or things like that. But I think in the main, what your guys' data is indicating is that that's not true. And if anything, you know, at least under certain sets of conditions, the Apt2 mutation provides the plant with an advantage. You know, I saw resistance to sulfate. I saw resistance to salt stress. I saw, in general, better seed production under normal growth conditions from the Apt2 gene. So, you know, I'm a little bit more hopeful that that this gene might actually prove to be useful someday. So I'm about to move from Arabidopsis to a project where I essentially do the same thing in tobacco as we did in Arabidopsis. The reason that I'm making that shift is that tobacco is a real crop. Obviously, it's not used for food, but it's easier than most crops are to manipulate genetically. And so you better believe that one of the first experiments that I'll do in tobacco is to find the tobacco copy of Apt2 and mutate it in the same way that we did in Arabidopsis and see what the results are. And if the trends from tobacco and Arabidopsis are the same, then I'll switch to a real crop like soybean, which is very difficult to manipulate genetically. But if I have a good set of data which suggests that it's worth doing, then I'll move ahead with that. You guys are contributing to that, so thank you for your work. So the last thing I want to do is let you guys know how terrific your presentations were. They were all well organized, and your professionalism was very impressive. I appreciate that. Congrats on the good job. So the last and final thing that I'll say is that there are obviously a lot of good students in the crowd. I hope that some of you will consider Virginia Tech at some point. If not for undergrad, I know I'm in Cavalier country, but if not for undergrad, maybe for graduate school. I'm always looking for good graduate students. So 6 years down the road. Take care and good luck.
So where do we need to bring these plants to?